Okay, well, good afternoon, everyone. We're going to change a little bit. I'm going to show you a little bit of the data we've been working on in my lab for the past 3 years or so on α-crystallins and different retinal disorders. First of all, for people that might not be aware of what crystallins are, very short introduction. They're called crystallins because they were first described in the lens. And they were thought at the time to be tissue specific. There are two main crystalline gene families, the alpha, that are small E-chalk proteins, and the beta gamma, that are originally believed to be structural proteins, but now there's more and more evidence that they have different functions in different tissues. Like I said, they were first discovered in the lens, and for good reason. They represent 90% of the water soluble in that tissue. While they were thought to be just there for transparency and refractive properties originally, now we have more and more evidence that they're expressed in other tissues, including the retina. And there has been different papers that came out from our lab and others showing in retinal diseases such as endophthalmitis, uveitis, ischemia. In our lab, we've been focusing on diabetes, on which I'll spend a bit of time, but also optic nerve injury and retinal trauma or light toxicity. So first of all, the retinal trauma. One of the models we've been focusing on, that's awesome, is retinal detachment. And the gamma-crystallins seem to be the ones that are the most affected by this model. So as you can see here, in 1 day, 3 days, and 7 days post-detachment, there's a little bit of induction of alpha-crystallins, somewhat some variability with the beta, but the gamma-crystalline pattern is very interesting with significant reduction as early as 1 day, and it's conserved over time with 3 and 7 days post-detachment. The role of that reduction is still to be understood, but this is one of the first, if not the first, evidence of an extra lenticular role of gamma-crystallins, and especially in the retina. Why are we interested in crystallins? Well, the reason for that is based on the evidence that have been developed in the lens. We know that alpha-crystallins specifically form oligomers and have chaperone activity. There are evidence that they could be involved in regulation of inflammation, as well as regulation of cell survival. Specifically, I'll get into that in just a minute, regulation of apoptosis through interaction with apoptotic regulators. More recent evidence also suggests that some of them, alpha-B and beta-B2 specifically, could be involved in regeneration and guidance of neurites. And finally, again, in the past 3 years, beta-A3A1 as well as alpha-B have been shown to have a potential role in vascular network and angiogenesis regulation. Now, in our case, we've started to be interested in them when we did a retinal proteome analysis, and which demonstrated that there was a subset of those proteins, alpha-A, beta-A2, A4, B2, B3, and gamma-B, among others, that were induced and dramatically induced up to 40-fold increase in diabetic tissue from rat animals. Now, what we did consequently was to actually check if this was seen also in human patient tissues. And we've collected a series of human donor tissues and looked at all different parts of the eye. You can see on the left-hand side vitreous, RP, and optic nerve tissues from non-diabetic, diabetic, and diabetic with PDR. And there was no variability, no changes for either alpha-A or alpha-B-crystallin in those human donors. Now, what was very interesting is when we looked at the retina itself, and we segregated between peripheral retina, central retina, and macula. And when we did that, we saw an increase in the diabetic and further induction in the diabetic with PDR. And that was true for alpha-A in peripheral retina, central retina, and macula. What was very interesting to us also is that for the first time we had actually some different effect on alpha-A and alpha-B-crystallin. when we also saw in the peripheral retina increased expression in diabetics and the diabetics with PDR, in the central retina and the macula, the increase wasn't seen in the diabetic alone, but only at later stages of the disease. Now, are the crystallins really basally neuroprotective? And if so, why are they increased in diabetes and we still see neuronal cell death? So one of the ways we've tried to address that is, as most people ask the question, well, if we take them out of the picture, what happens? So we've been working with the alpha A and alpha B crystallin knockout mice, as well as the double knockouts. And what you can see here is that the gray and black bars that corresponds to alpha A and double knockout show very significant higher level of cell death at 8 and 12 weeks of diabetes than the wild type and the alpha B-crystallin. Interestingly, the alpha B-crystallin did not show any increased cell death at all the way to 24 weeks, which is when we really saw the dramatic increase in the wild type as well. Interestingly, at that time, the level of cell death in the alpha A crystallin knockout mice completely dropped. Now, this time point is preliminary. It's only one cohort of animals that we've done, so very cautious with this. But this is interesting, and we're repeating that as we speak. So what we try to do as well is correlate that with a longitudinal study of the retinal thickness of the retina in those animals. And we saw somewhat similar to what we saw with the cell death, a thinning of the retina in the alpha A-crystallin knockout as early as 2 and 4 weeks, a thinning that we actually didn't see, well, much more important than what we saw in the alpha B knockout or in the wild type. On that graph, we don't have the double knockouts because they develop cataracts so early that we can't look at the retinal thickness. So now we have this cohort of animal that we take through all the 24 weeks, and we're actually doing the histology on those guys at the different time points. So that suggests that lack of alpha A-crystallin at least, and concomitant lack of both alpha A and alpha B-crystallin leads to increased sensitivity to diabetes. Now, how does that match with what we've seen, and why increased crystallins then is not protecting the retina in diabetes? Well, this is how we looked at that. There was evidence in lens epithelial cells that crystallins interact with pro-apoptotic proteins such as BAX, and that this interaction prevents BAX from going into the mitochondria and forming the apoptopore. So we knew that alpha-crystallins were highly induced in diabetes, and we had also shown, I'm not showing you here, that BAX is also increased in diabetes. And what we did here is just look at the interaction between the two proteins, well, the three, I should say. And so while their level of expression are fairly low in controls, they interact together. We could pull down BAX and alpha B-crystallin together and pull down alpha A and BAX together in controlled conditions. Interestingly, while their level are highly induced in diabetics, the interaction is almost completely lost. So there's something happening to those crystallins during diabetes that prevent them from interacting, and that might explain why we do see this increased cell death in diabetes. And one of the things that we think is regulating this is post-translational modifications. We've recently published that in addition to the fact that their level of expression is highly increased in diabetes, there is also higher levels of phosphorylation of alpha-B-crystallin on serine 59, 45, and 19, phosphosites that are known to regulate the chaperone and interaction of those proteins. And we've looked in culture mutating those different sites, and what you can see here is that when we do a triple D mutant or triple alanine mutant mimicking the phosphorylation or blocking the phosphorylation, you can see that it's similarly to what we've published in diabetes, where we saw that a great amount of alpha A and alpha B-crystallin ended up in the non-soluble fraction of diabetic samples. Mutation of those sites rendered those proteins either completely soluble or very insoluble. So suggesting that The effect of diabetes on the function of crystallins could be associated to this effect that we see on the phosphorylation pattern, leading to loss of solubility and other loss of function, basically, of crystallins. Now, along the same lines, just a very quick side note, we're working on other crystallins, and there are some parallel in their regulation. Here, I'm just going to show you an example of a triple mutant that has been identified in a family of human patients with glaucoma and cataracts. And it's a triple mutation affecting beta-B2-crystallin. And when we express this beta-B2-crystallins, if we express the wild type, we see it nice and diffuse in the whole cytosol and even a little bit in the nuclei. And when we express the mutant form that we've identified in those patients, we see that they form those inclusion bodies and those aggregates, and that could be associated with, again, a loss of function. Okay, now this is for those. We have more knowledge about αB-crystallin because of all the work that's been done around those, but α-crystallin we don't know nearly as much, and we don't have as much tools. So we asked the question, especially since the α-crystallin knockout is much more sensitive to diabetes, could there be also some similar type of regulation? Now, no phosphosites have been identified in α-crystallin, or at least not described to the extent of what's been described for αB. So we used a different approach with mass spectrometry, and what we've seen is very interesting. We've identified a site on serine 148 that's highly phosphorylated and exclusively in diabetes. Now we're only starting working on that, making those mutants and identifying the impact that it has on the function of the proteins. One of the things we're interested in as well is locating those changes. And so we're working with Dr. Kevin Shai at Vanderbilt to using the approach of MEL imaging to localize specifically those phosphorylation and those post-translational modifications. And the way it's done is very quickly. You get an image of the tissue that's been laid on the matrix, and then after you've shot at the tissue, you can actually reconstitute the heat map corresponding to the peptides identified or the full proteins identified. And in this case, then you can superimpose them, and you can localize the different layers, and that gives you a direct indication of where those different PTMs are localized. Okay, so now what about other contexts, other diseases? Very quickly, we have been interested in AMD, and one of the reasons for that is that crystallins are known to be, αB-crystallin especially, is known to be present in tissues from patients with dry AMD. And we've, like Dr. Chen said, there is no really good model of AMD. One of the ways we've been looking into this is sort of similar, sort of a quick way of looking at this is the light toxicity model. And interestingly, what we've seen is looking at specifically at the outer retina. In wild-type animals subjected to light damage, in those conditions, we lost about 50% of the outer nuclear layer. And when looking at the different knockouts, you can see that the layer is almost depleted. And similarly, in the superior half of the retina that is known to be less affected by this process, we go from about 80% to 40%. This is just a representation of the numbers, but basically suggesting that both alpha and αB-crystallin renders mice more sensitive to light toxicity model. So in conclusions, we have shown in humans and other models that crystallins, protein overexpression is a hallmark of diabetic retinopathy, that it's upregulated in cell-specific manner. I haven't shown you that really. But that it has a neuroprotection potential, but that potential is destabilized by diabetes, and that part of that might be regulated through the phosphorylation of those proteins. Now, in other diseases, there are different crystalline subsets that are affected, like I showed you with the retinal detachment, for example, or I didn't show you, but with the light damages similarly, and that there's also a cell specificity of that. There's still a lot of work to be done on that aspect. And last but not least, thank different people that have participated in that work. Mandy has done a lot of work with the animals originally. Caitlin has done all the OCT work. And then Jen is doing all the cell work. David Zaks, collaborates on the retinal detachment project, Christian on the light damage, and Kevin, as I said, on the multi-imaging. And I'd like to thank the sponsors and thank you for your attention.
When you're performing the OCTs, how are you actually measuring the outer thickness for the same area, because you have to do it over time? So how do you actually do that?
Well, we get images of the whole scan. We can do B scans, and then we can keep the angle on those mice. So really, the imaging process is very standardized, and it's easy to actually go back, and you can take the measurements at different sections, different places. That's how we do. So for the wild type and the αB-crystallin knockout, we can actually follow them over time very long, and that's very, there is some variation over time just due to age, but the variation inter-individual is intra-individual is very small.
Thank you very much. Thank you.